All right, so now we're going to talk about the viral structure, so the structure of various types of viruses. In general, virions, or virions as they're also called, are the viral particles, are small and cannot be observed using regular light microscopes. So they're so small we can't use our regular light microscope. We can't get an image like in A here on a microscope that we could use in a laboratory. They're much, much smaller than prokaryotic and eukaryotic cells. This is an adaptation that allows the viruses to infect these larger cells. So viruses are so small that then they're able to infect prokaryotic cells, like bacteria, for example, or eukaryotic cells because they're so small. And the size of a virion or virion can range from about 20 nanometers up to 900 nanometers for large viruses. However, now they're finding these new giant viral species, such as Pandora virus and Pithovirus. And so those are so large that they're getting to be about the size of a bacteria cell. All right, so we're going to scroll down. We'll take a look at this image again in just a moment, but we're going to scroll down and go through the words here. Perhaps you remember this image from the first chapter or two. Look at the different sizes of things. So we can see here that when we're talking about viruses, we're talking in the very, very small range here, much smaller than a bacteria cell, for example, or a plant or animal cell. So what we're going to see that these viruses are so small that they can infect all of these different things like bacteria cells, plant cells, and animal cells. All right, so here in 1935 rather, after the development of the electron microscope is when we see the first viruses. So we have to be using something like an electron microscope in order to even see them. Wendell Stanley was the first scientist to crystallize the structure of the tobacco mosaic virus and discovered that it's composed of RNA and protein. So unlike bacterial cells, unlike bacteria cells, and unlike eukaryotic cells, we don't see all of those different types of organelles. We don't see the cytoplasm and the same type of arrangement that we see. We see very similar arrangements within plant cells and animal cells, both being eukaryotic. And then even when we look at prokaryotic cells, we still see cytoplasm, we still see nucleic acids, we still see some sort of organelles and an outer membrane, but viruses are very different than that. We don't have a cytoplasm, for one example, but we do have these things to maintain a virus. Again, it's not considered life, but it is something in between. So what they saw was in this tobacco mosaic virus that it was composed of RNA and protein. In 1943, he isolated the influenza B virus, and then that is what contributed to our flu vaccine, the beginning of a flu vaccine. Stanley's discoveries then unlocked the mysteries of nature that have been puzzling scientists for over 40 years. So he did end up getting the Nobel Prize in 1946 for his discoveries. So as a result of this research into viruses, we now know that they consist of a nucleic acid, so either RNA or DNA, but never both. Okay, so in our eukaryotic cells and in our prokaryotic cells, what we see is that they have this core, either in a nucleus in a eukaryotic cell or in a nucleoid region in a bacterial cell, for example, and they're composed of DNA. And then they go through the process, if you remember, in eukaryotic organisms, going through the process of taking DNA and making RNA to then make proteins. In the case of a virus, we see nucleic acid as well. However, it's either RNA or DNA, and we'll take a closer look at that in just a little bit. So they have either a nucleic acid, either RNA, or they have a nucleic acid, either RNA or DNA in the center, and then it's surrounded by a protein coat called a capsid. And that's the most basic structure of a virus, just some sort of nucleic acid surrounded by a protein coat called a capsid. The interior of the capsid is not filled with cytosol, like in other cells that we discussed, but just those bare necessities, meaning genome and certain very specific enzymes that are needed to make the new virions. So each capsid is composed of subunits called capsomeres, made of one or more of a different type of capsomere proteins, and then they're going to interlock to form this coat, this outer region here that's going to protect those nucleic acids. There are two categories here of viruses based on their composition. There are viruses formed from only nucleic acid and capsids. Those are called naked viruses, or non-enveloped viruses. And then there are viruses that have a nucleic acid, a capsid, and then surrounding that capsid they have another lipid layer that's called an envelope. And these are enveloped viruses. So we have non-enveloped viruses and enveloped viruses. A non-enveloped virus is just nucleic acid surrounded by a protein coat called a capsid. And then our enveloped viruses are nucleic acids. Then they have that capsid just like the non-enveloped viruses. And then they have an additional layer. So this lipid layer that's like a phospholipid membrane. So the viral envelope is a small portion of phospholipid membrane that is obtained as the virion or virion buds from the host cell. And we'll look at that in a little bit more detail in just a moment. But when the virus is going through the process of replication, when it's going to burst out of the cell or bud out of the cell, it's going to end up taking some of this phospholipid membrane, that double membrane that we have on the outside of all of our cells that is our barrier between one cell or the other. And it's going to take some of that with it. And then that's going to be its viral envelope. It can actually be either intracellular or cytoplasmic in origin, meaning either the virus can direct that it gets an envelope, that would be intracellular, meaning that once it's in the Golgi apparatus, the rough endoplasmic reticulum, it can grab some phospholipid membrane from those organelles and then go out. Or it can be cytoplasmic in origin, meaning it can be from that process of budding, where it's going to bud out and grab some of the outer membrane with it. Also, what we see are some spikes on viruses, so extending outward and away from the capsid. On some naked viruses and enveloped viruses are protein structures called spikes. So we can also see spikes on the outside of some of those viruses. At the tips of the spikes are structures that allow that virus to attach and enter a cell, like the influenza virus, the hemagglutinin spikes, and the neuraminidase influenza virus spikes that allow them to detach from the cell surface and then release new virions. So you don't need to know these two specifically, the H and the N. What you should understand is that they have these spikes off of the outside of their either capsid or their envelope, and then those spikes are what allow them to attach to the cell, get into the cell, and do their thing. So it's just mentioning here that the influenza virus, perhaps you've heard of H1N1 or H2N2, those are referring to these spikes on the flu virus. So let's take a closer look at some different types of viruses then. So we have different shapes of viruses, and that's due to the shape of their capsids. So they can be either helical, polyhedral, or what's called complex, or basically the rest of them. So the helical capsid is like the tobacco mosaic virus. The tobacco mosaic virus is spoken about a lot because that was the very first virus that was kind of investigated, and we talked about that already, but it's a good basis for viruses because it was looked at for so long and was the first to be looked at. So the tobacco mosaic virus is a naked helical virus. So naked meaning it doesn't have the envelope, and it's helical, and there are actually some images, maybe not here, but down here, of our helical virus. So, for example, the tobacco mosaic virus, you can see it's kind of long tubes is what they look like, and you can see that there's the illustration of it here. And they are, you can see the DNA in here, or RNA, depending on what the nucleic acid is, and then this is their capsid out here, the yellow kind of lumps or proteins. So this is our helical in shape virus. Also, the Ebola virus is a helical virus, but it is an enveloped virus, so it has an envelope. So we have our TMV, or tobacco mosaic virus, which is a naked helical virus, and our Ebola virus, which is an enveloped helical virus. It's cylindrical or rod-shaped, as we saw, with the genome fitting just inside the length of the capsid. Then we have our polyhedral capsids, form the shapes of poliovirus and the rhinovirus. And they are a nucleic acid surrounded by a polyhedral capsid. So it's in the form of an icosahedron. An icosahedron, or an icosahedral capsid, is three-dimensional. It's a 20-sided structure with 12 vertices. So basically it looks like a soccer ball. So we have these sides that are coming together, and you can see it here with this rhinovirus. You can kind of see it. It ends up looking like a soccer ball. You've got all of these different pieces, and actually if we look up here, you can see a little bit better on our bacteriophage here. This would be like an icosahedral. You can see a little bit more three-dimensional shape here. So you've got the different sides, and then they're all kind of stuck together. This right here, this B, this whole thing is not an icosahedral or a polyhedral. I'm just using this one portion of it as an example. This would be a complex shape, actually, so we can move into the complex shape now. So a bacteriophage, which is what this is a virus that infects a bacteria, and this would be an example of a complex virus. So it actually has this icosahedral top here with the nucleic acid in the inside, and then it has its capsid around here, but then it also has this tail region. So we have a sheath here, and we have tail fibers, and we're going to take a little bit closer look at that in just a moment. But this would be an example of a complex virus because it has more than just the helical shape and more than just the polyhedral shape as well. All right, so both helical and polyhedral viruses can have envelopes. And then here, viral shapes seen in certain bacteriophages or bacteriophages, such as T4 phage and pox viruses like vaccinia, may have features of both polyhedral and helical viruses, and so they're considered complex. So you saw that there's the top portion, which was the polyhedral portion, and then it had that sheath part, which was the helical portion, and then that gives it a more complex shape, and it's put in that category. So it showed us here, or it's saying here, that the genome is in the polyhedral head, and then the sheath, as we mentioned, has the tail fibers and the tail pins that help the virus attach to receptors in the host cell surface. All right, and then actually here you can see another example of that, another example of a complex, which is not the bacteriophage, but is just a varying type of virus here. So you can see the nucleic acid, you can see that this one has an envelope here, and then the capsid surrounding the nucleic acid. So it's not an icosahedral shape and it's not a helical shape, so it's just classified as complex.